This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 244. Recorded on June 10th, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Michigan, Michele Swanson.
Hello, good to see you.
You're not in Ann Arbor, so I don't know where you are, but Michigan.
In the woods.
In the woods of Michigan. Good to see you. Where I understand it's pretty cool. That's nice.
Oh, no, I mean, it's temperate, right? 75 degrees. It's perfect.
Very nice. Also joining us from Small Things Considered, Elio Schaechter.
Well, hello there. Well, it's another 75 degrees.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. But we have swamp weather today. It's probably, let me check. Right now, it's 88 degrees with a chance of thunderstorms, and the humidity is about 100%.
Oh, that's miserable.
Charleston, yes. South Carolina. I presume you have air conditioning at home, right?
Oh, yes. It's the only thing that allowed the South to really recover.
Well, I still have never been to Charleston, so one day to get there when we recover from this pandemic.
And the food is great, no matter the temperature, right, Michael?
That is true.
Yeah, that is true. Really good. Yeah.
Though we haven't experienced the restaurants, they've been writing in the newspaper how much of a struggle it is to get wait staff and line cooks and things like that. They really lost a lot of folks due to needing to find work after when the pandemic started. It's probably no different in any other community.
Yep. It's hard to get people back to work. Yeah. All right. Well, the weather is variable, but what is not is the science. And here we have really good science on TWiM for your listening pleasure. So let's start out with we're going to continue our food arc with Elio.
Well, hello. The paper I'm going to describe is called Bacteria Associated with Human Saliva are Major Microbial Components of Ecuadorian indigenous beer, chicha. I'll explain all of that. It was published quite a while ago in a journal called Peer Journal. The authors are Ana Freire, Sonia Zapata, Juan Mosquera, Maria Lorena Mejia, and Gabriel Trueba, who is a professor at the Universidad de San Francisco in Quito, Ecuador. He is published a great deal on essentially based on sequence analysis of natural samples. So we're talking here about a drink called chicha. Let me start out by saying that alcoholic beverages typify human societies. Think about champagne in France, scotch in Scotland, moonshine in Appalachia, moonshine in your part of the world.
Bourbon in Kentucky.
Bourbon. So every society seems to have a variety of alcoholic beverages, but some are kind of unique. So chicha, that's C-H-I-C-H-A, it's not a Spanish word, it's probably a Quechua word or something like that, is typical of the western regions of South America, namely Peru, Ecuador, and Colombia, typically, although it spills over into adjacent countries. So the main artisanal alcoholic drink, chicha, is made by the native people, was made historically by the Incas, and where it was appreciated as kind of a royal drink. Anyhow, it is the product of fermentation of one of various grains, mainly corn, but also non-grains like cassava, that's manioc, which is a carbohydrate, polysaccharide-containing root, and even the fruit of a certain palm tree called chonta. Anyhow, so a lot of things I use to make in chicha. What typifies chicha and what's going to get your attention pronto is that to make chicha, you have to chew the grain or whatever it is. So you got to chew it for a while and spit it out into a pot and then let the pot ferment due to natural occurring fermenting organisms.
So
Okay.
that's the fermenter who has to do that. It's not me, the consumer. It's the fermenter.
That's right. Okay, so typically the chewing of chicha is done by women. As I say, in Inca times, it was done by the virgins of the sun, whoever they were. They were in charge of doing this chewing. Now, lest you think that this is disgusting, it is not, because it is consumed by a large variety of people. And after it going through the fermentation, things have changed significantly. So I used to live in Ecuador. I spent my teens there. And as a teenager, I did not drink chicha. I was abstemious. But on a trip back, I went to a chicheria, which is a place where not only do they make the chicha, but also you drink the chicha. And I had a glass of chicha, and it was okay.
In full knowledge of how it was prepared, you drank it?
Oh yeah, I knew very well how it was prepared, but I drank it anyhow. And it's sort of a thick, yellowish drink, which tasted okay. I'm not going to become a chichero, a big chicha fan, but it was okay. And anyhow, it was worth it, since it's used on a wide scale. With different substrates being used. The study by TWiM's laboratory, which is a very up-to-date laboratory, was well worthwhile. So what did they find? They found that, like others had found before, in fact, they extended this to find that the main bacteria that are found there are Lactobacilli, which in chicha. In other words, after the fermentation, by the way, only takes 2 or 3 days. So it's not a very high alcoholic content thing, about 2-3% alcohol, much less than beer, leave alone wine. So, but if you drink enough of it, you get enough alcohol. So if you get over your qualms and you drink it, you're going to find that the microbiology consists mainly of bacteria which are derived from the human mouth. Many are lactobacilli. The main ones are lactobacilli, and this is done by using 16S ribosomal RNA. Okay, I think Michael will have something to say about how widespread that can be, how easy it is these days to do 16S. I'll leave it for him. Anyhow, by the way, today's whole TWiM is South America oriented. You'll hear a little bit from Michael about that. You'll hear lots from Michael about that. Anyhow, TWiM found that the most common, as I said, the most common bacteria are lactobacilli. But in addition, he found something which was not expected quite, namely 2 kinds of streptococci, Streptococcus salivarius and Streptococcus mutans, which are both potential pathogens. Strep mutans is an important agent of dental caries. It may be the main agent of dental caries. That's not entirely clear, but anyhow, finding Strep salivarius and Strep mutans is of some significance because it says that the chewing of the corn is not for free. You get potential pathogens.
They also, if I could point out, Elio, in just under 4% of the population, they found Streptococcus pneumoniae, better known as pneumococcus, and that's for sure a pathogen.
Yeah, right. That's right. So anyhow, although chicha is drunk by millions of people with impunity, apparent impunity, it may not be. So be careful if you go to Ecuador, Peru, or Colombia, and you're offered a glass of chicha, go ahead and drink it. But be aware that you're taking a small chance that you may acquire a potential pathogen. And that's about the story. It's a nice article in the sense that it is thoroughly done. It is well done. And they sampled 4 different kinds of chicha. Some were chewed cassava, some were this palm seed called chonta, and some were corn. And these were different in geographic regions. 3 of the samples were from the Amazon, one was from the highlands. What they didn't do is to determine the amount of alcohol in the samples after fermentation, but it's probably irrelevant because it's not very high. So there you have it. It's a study that takes advantage of the availability of modern techniques to sequence anything you want. And this is done right. This is done correctly. The extraction of the DNA was done correctly. The polymerase reaction was correct. Everything is very on the up and up, and this is it's an elegant study.
I agree. One of the other controls I was especially interested in is they also looked at the microbiome of the drink that was made with mashed chicha. So this wasn't chewed by people, but it used the same plant as a substrate. And they still found human microbiota bacteria were fairly common in those as well, and they speculate that it might be from just the traditional practices of where the fermentation is done. Maybe some of the buckets or the containers that they use have a biofilm, and they even speculate, because this has been going on, for eons, that some of the streptococcal strains may have actually adapted to grow in this drink or on this plant. So I thought that was interesting.
Yes, it is. You're right. And there are a whole lot of other bacteria too. Some I never heard of, Weissella, Confusa, and others like that. So it's quite a collection of bacteria. But as I say, Lactobacillus, Lactobacillus plantarum are among the most common one. Oh, they also found yeast. They found fungi, and the most abundant of the fungi among them was Saccharomyces cerevisiae, the common yeast. So that's about the story.
Elio, this is a small time beer, right? This is not a big business. It's just
not Budweiser.
Yeah, I mean, it's small time. It's small time production, right?
We call that craft. Excuse me. That would be a craft drink.
Yeah, right.
Yeah, Wisconsin and Michigan, they call it craft.
Artisanal.
So there's no desire to, for example, replace the chewing with known microbes, right? Nobody cares about that.
That's easier said than done. In my misspent youth, I worked on developing inocula to transform silage into a better food for farm to table processes. Yeah. And it's really complicated in the sense that what I learned is that there are many different strains of Lactobacillus plantarum. 16S is the same, but there are many unique strains of Lactobacillus plantarum, and who's there really determines whether or not that forage, and this is really a forage process where you're chewing, you're adding salivary amylases, all of the salivary processes where when we begin to chew, we release enzymes from our saliva that begin to digest the starches, which makes it more available for alcohol fermentation. And if any of our listeners frequent the TV show Moonshiners, they'll learn that what the Moonshiners do is they make sure that the surface area to mass ratio is really high for the ground up carbohydrate. They typically mash in with corn or any of the other carbohydrate processes. But one of the things that I learned watching Moonshiners is that if they malt the grain, where they allow the grain to sprout, it effectively begins to break down the starches.
Right.
And that's effectively what gives Scotch its different flavors and what gives Kentucky bourbon its enhanced flavors.
Well, and it releases the sugars then that the
Yes.
microbes can use to grow.
Because you got rid of the polymer and you've gotten a sugar. And I think that's principally why the Incans effectively did it, because you could get more alcohol out of the process.
I didn't say that human saliva contains amylase,
Yes.
which is an enzyme that breaks down starch into sugars. So that's probably the main point of doing the chewing. By the way, they don't have to chew for very long. They just have to mix it with saliva. And also, I read someplace, don't ask me where, that this is not only done with chicha, but also with sake.
Yes.
You can take soybeans, and you coat them with human saliva before you break them up. And this adds amylase and lets the sake fermentation go on. So this is not just an invention of the Inca Empire.
Yeah.
Well, I think the Incans were probably one of the first races to experiment developing the microbiome to a productive activity. You know, this was a productive activity. They made an inoculum by having the sun virgins chew it, and effectively they ate a common diet so they could have reproducibility. So it's actually a quite fascinating story. And if you think about the challenges we're going to have in developing inocula and probiotics and prebiotics to give to people to improve health, this seemingly fun paper about making a beverage really is an object lesson in how our ancestors did it well. And it's literally been past the test of millennia.
So let me say that in the introduction of that paper, they quote a paper by Carrigan et al. That says, the domestication of fermenting bacteria and yeast predates the domestication of animals and plants.
That's right.
Ancestral hominids adapted to metabolizing alcohol long before the Neolithic period. So this goes back a ways.
And the benefit was that that reduces the risk of foodborne illness, because the fermentation process changes the pH, and then it's a less fertile media for bad bugs to grow.
There's one year at ASM, I heard a talk by a microbiologist whose specialty is ancient alcoholic beverages. And what you do is you can go into, you could find old pots, thousands of years old, and there's a little bit of the beverage left, and he takes it out and tries to figure out what's in it, and then goes backwards to try and reproduce it. And he actually works with a company that sells this stuff.
Wow.
I think we did an episode on TWiM, but I can't.
I'm trying to remember. It sounds familiar.
And I bought a bottle of it just to see what it was like. I think this was one of there were Chinese fermented beverages and the Egyptian, but it's quite interesting. I mean, he has made a career. He's an academic, and he's made a career out of figuring out what's in this and trying to figure out how they made it. It's quite interesting. So people like to get tipsy right from the start,
From the beginning of time.
Or avoid waterborne infections.
How does that work, Michele? What do? You make beer and you soak your meat in it? Is that what happens?
Just that water may not be safe, but once you ferment it, then and then you get preservation. You can store over the long winter or whatever.
Lowers the pH and all of the above.
Yeah.
So that's the excuse for drinking beer in the morning, right? You say, I can't drink the water. It's bad. I have to drink beer.
Well, actually, it's why the men around Snow's famous pump didn't develop cholera. It was only the women and children who developed cholera because the women and children drank water, whereas the men only drank beer.
Interesting.
There you go.
They love these snippets. All right. Thank you, Elio.
I have to make one more point. There's a store near me that instead of going to a farmer's market, it's a farmer's store. They bring produce in from the local, and you can go and buy produce. And they have a large banner that's like, know your farmer. Mm-hmm. And certainly if I was buying chicha, I'd want to know my brewer.
Your chewer.
Yeah.
Your
Oh, you
chewer.
Anyhow, I thought that this article by the TWiM Live was a good indication of what you can do,
Yeah.
Out in the field. And
Yeah,
it's
cool.
nicely done. It's a well done paper.
Yeah, I like it. Thanks,
The
Elio.
emphasis is on papers like this is on doing it right and interpreting
Yes.
it right. So nicely
Okay.
done.
All right, Michael, what do you have for us?
So we're going to continue on our South American journey with a paper from Applied and Environmental Microbiology entitled Unveiling the Virulent Genotype and Unusual Biochemical Behavior of Escherichia coli ST or sequence type 59. And it's by Demelo Santos, Fuega, Esposito, Cardoso, Fernandez Santos, Barchalelo Valente, Santos Nito Gales, Lincolpon Silva, and Gomez. And they are in the discipline of microbiology in the Department of Microbiology and Immunology and Parasitology in the Paulus School of Medicine at the Federal University of Sao Paulo in Sao Paulo, Brazil. So it's a really neat story, and it's got many object lessons for us to think about. In the beginning, when I learned about E. coli in the last century, I was taught that it was operationally defined as a gram-negative, facultative anaerobic, paratrichously flagellated rod that was a mixed acid fermenter, but specifically produced acid and gas from the fermentation of lactose, thereby earning it a place in a unique category of the enterics known as coliforms. And if you're quickly looking for your list of coliforms, they are Citrobacter, Enterobacter, Hafnia, Klebsiella, Escherichia, and the last one is Serratia. Why coliforms are important today and what it has to do with anything is what we're doing across most of the Northern Hemisphere, namely summer heat, where folks are cooling off at beaches and pools, with coliforms being commonly used as an indicator of the sanitary quality of foods and water. Coliforms are diverse in the niches that they occupy. They can be found in aquatic environments and soils, on vegetation, in people, in animals. They're universally present in large numbers, in fact, in the feces of warm-blooded animals. So you get the idea that if someone asks you if bears really do what you think they do in the woods, you can offer that I have a test for that. And in fact, there's actually a test. It's an ISO 9308 test that can actually look at water quality and answer the question, do bears do it in the woods? Because you can enumerate the number of E. coli and coliform bacteria associated with that stream water that you're drinking while you're camping. So today's story, as you can probably surmise from the title of the manuscript, is going to upset this dogma and revolves around an extra-intestinal pathogenic form of E. coli that they abbreviate XPEC. Well, they don't actually abbreviate it. It's actually the nomenclature of the field. And reviewing, there are many different types of E. coli out there, with the routine categories of our strains responsible for enteric diseases, including enteropathogenic serotypes, which are often referred to as EPEC strains, enterotoxigenic E. coli, or ETEC strains, which make a toxin that's secreted that causes the effect, enteroinvasive E. coli, which are really bad because they invade our tissue, the enterohemorrhagic E. coli, the ones that we don't want associated with our meats because they make a hemorrhagic toxin, enteroaggravated strains, or EAEC strains. And today we introduce the 6 variant type to your nomenclature or repertoire to worry over, and that's this extra intestinal pathogenic form, or XPEC. Now, this microbe has all sorts of virulence determinants, how the microbe is able to cause disease in the host. And if you will, the genes are components of a term I don't think we've ever really used on TWiM, but it's in the literature, termed the virulome. And the virulome is, I'm going to use this paper to introduce this concept to our thinking. And I'm going to use the data they present to really highlight these genetic traits within bacteria that enable it to be a pathogen. And so this is an extra-intestinal pathogenic form. So that means the pathology that it manifests is not associated with the intestine. And in point of fact, this microbe was isolated from the bloodstream of a 91-year-old woman who was hospitalized due to gastrointestinal bleeding, and they recovered the pathogen from a blood culture. Routinely, they bleed you while you're in the hospital to look to see whether or not there are bacteria in your blood, and they isolated this as the sole organism from this 91-year-old woman who was suffering from an infection. She ended up going into septic shock during her hospitalization course, and unfortunately she passed. She died. This by itself is not unusual in that XPEC strains are one of the leading causes of human and animal infections on a global scale. They selected this particular strain of E. coli from this human blood culture because it displayed an unusual, atypical light cream colored colony when grown on a chromogenic agar plate. Now, chromogenic agar plates are unique in that they anticipate that the microbe that you're trying to characterize is effectively going to consume a particular substrate and then effectively wave at you from the plate, alerting to...
Don't make him sound like there's something new. That old as the hills.
Oh, no, they're as old as the hill. Absolutely.
EMB.
EMB, a McConkey agar. There's a whole list of selective and differential media that clinical microbiologists have used literally now for over 100 years to exploit this. Now, this particular microbe was unable to produce beta-galactosidase, the enzyme responsible for one of the canonical traits of E. coli, namely the ability to ferment lactose. Didn't have any beta-gal. In addition, it didn't have any beta-glucuronidase, which is another enzyme that E. coli has that is used in waste and water analysis. Now, an absence of the beta-gal enzyme would immediately suggest to someone, and again, I always think back to my introductory microlab days, that if it wasn't lactose positive, it wasn't E. coli. Right. Well, this is not the case here because this didn't have lactose. And in fact, the genomic analysis of the strain showed that it is indeed was E. coli. And in point of fact, it was an especially virulent form of E. coli in the fact that it killed a woman and it had all of these genes associated with the virulome, genes implicated in the ability of the microbe to cause disease in a human host. And the genes that it had, and I'm going to go through them, it had the PAP gene, which is a chaperone usher-type fimbrial appendage found on the surface of many E. coli. And as you can imagine, the PAP facilitates its ability to attach to our tissues. It also had KPSE, which is a virulence protein involved in K for capsule synthesis, the capsule polysaccharide export intermembrane protein that facilitates the deposition of capsule on many E. coli.
And just to point out, the capsule equips microbes to avoid being eaten by neutrophils, for example.
Yes, the evil phagocytosis process that our immune system has cooked up. It also had CHU-A, which is an outer membrane heme receptor. And as you can guess, this scavenges the precious resource iron. It also had FU-A, which is the pesticin receptor, which is another siderophore, a public good that bacteria employ, and again, enabling it to scavenge iron, because in a human host, iron is locked up tighter in this vault called transferrin, and it's designed to scavenge iron. And finally, it had YFCV, which is an uncharacterized fimbrial-like adhesin protein. Now, the nomenclature of the discipline is the virulence potential of strains, or the virulome, if you will, can be assessed using molecular criteria described for the definition. So this is a canonical definition of XPEC. And if an XPEC strain has at least 2 amongst the 5 specific virulence factors, PAP, SFFA, AFA, DRA, and again, these are all these adhesin-like outer membrane stuff, as well as their uropathogenic potential by the presence of at least 3 genes. Again, CHU-A was one, FU-A was another, and YFCV is another one. So this organism is optimally designed to go extra intestinal as well as uropathogenic. Again, always go back to ask, why is this paper significant? In the introduction, the authors offer us that the tracking of these pathogenic E. coli is not a trivial in fact. And in point of fact, the primary focus of looking for how E. coli and all of these virulence factors have been spreading across the globe has been one of many One Health projects. Now, just to refresh your memories, One Health is this global consortia, collaborative, multi-sector, transdisciplinary approach looking at local, regional, national, and global levels with the goal of achieving optimal health outcomes, recognizing the interconnection between people, animals, and plants and their shared environment. We've learned this object lesson in spades this past year with COVID-19 and where COVID has come from. The CDC's One Health Office leads this agency's efforts in the United States and helps coordinate these activities from abroad. Where they have been watching E. coli especially is in its emergence of multi-drug resistant strains. If you will, the carriage of these resistome traits or antibiotic genes that degrade antimicrobials. And in fact, they are looking at the extended spectrum beta-lactamase and again, or the beta-metallo beta-lactamases. Again, this paper really drives home or highlights to me the value that clinical microbiologists who are curious have in helping the pathogenic microbiologists like Michele discern what might be going on respect to the virulence mechanisms. Ask yourself why any self-respecting pathogen would elect to get rid of its 2 species-defining canonical enzymes, beta-glucuronidase and beta-galactosidase. They don't have an answer for us, but they really highlight the loss of those 2 enzymes is really going to impact our ability to answer and assess whether our waters are safe, whether our food is safe, because for the most part, many of our systems use chromogers. They use broths. The broth that they used in this particular test was aqua teste coli, which is a broth that contains orthonitrophenolthiogalactoside, OMPG, which every molecular biologist probably has used once in their life. And OMPG is useful because it doesn't need the lac permease to get in to be cleaved by beta-galactosidase.
So, Michael, if I could just put a sharper point on this,
sure.
Especially now that we're in swimming season, our listeners have probably heard a certain beach is closed because of a high coliform count. There's clearly been a breach. There's some sewage getting into the water somehow. And typically how we monitor our beaches is using these simple plates. Are there any coliforms? Are they pink? Are they lactose positive? Then that is an alert. So now, as this paper will show, there is definitely a whole family of bacteria that are going to not be pink, but they're still loaded for bear. They can cause disease. So this is a real problem in our public health screening.
This is basically setting off alarm bells. I mean, this is the canary in the coal mine. And when the canary in the coal mine doesn't react to the toxic gases and die, this is effectively what's going on here. And they do this beautifully in their first figure. They show us what a positive chromogar plate should look like, and they show us what the isolate looks like. They also give us a negative control. So going back to what Elio taught us from the first paper, it's beautifully done. They illustrate what's going on. Then they show us this mug broth. And this mug broth is cute because it effectively assesses for beta-glucuronidase. Beta-glucuronidase looks specifically for fecal coliforms. It's beyond E. coli. It's found in streptococci as well, and there are fecal streptococci. And when I learned microbiology in the last century, you could actually determine if a bear was dumping in the woods or if a person was dumping in the woods by determining the coliform to fecal coliform ratio. And many, many places still do that sort of thing. But at the end of the day, this is still E. coli. And in fact, they go to great lengths to point that out to us, regardless of the unusual biochemical characteristics these microbes displayed. Both MALDI-TOF, which is effectively vaporizing the microbe colony and doing time-of-flight mass spectroscopy, where it's looking at the whole kit and caboodle of the microbe. And the BT Phoenix system, which is a redox indicator assessing for growth, no growth, that's frequently used in clinical labs to determine antibiotic susceptibilities, said, hey folks, this is still E. coli, irrespective of the fact that it's missing these two enzymes. They next wanted to know if this was a fluke to Sao Paulo. Was this a fluke just to this 91-year-old woman? And they next evaluated first by genetically sequencing this isolate, confirming that it was indeed E. coli. It had about 5,057,000 base pairs. The GC content was right. And they also offer that this strain belonged to the serotype O1H7 phylogenetic group F and sequence type 59. So we've spent a lot of effort characterizing E. coli because of the amount of disease it caused. As an aside, the O1 and O2 serogroups are frequently associated with human infection. So if you see an E. coli with an O1 or an O2, think human infections, especially in the bloodstream and urinary tract and meningitis. Recall that the O antigen is that side chain on LPS, on the outer membrane in E. coli. It literally sits out there and flaps out for our immune system to see. And in fact, there are at least 183 O serotypes. Hence the need for having these crazy license plate designations. Additionally, E. coli has a large number of flagella genes. Remember, it's paratrichously flagellated, so that means the flagella surround E. coli. And it has 53 possible genes, which means a different protein that it can swap in. So we now have 183 plus 53. And then finally, it was of the K1 designation, K from the Dutch or Danish for capsule. And E. coli has at least 80 different capsule genes it can swap in. So what this amounts to is that there can be approximately 1.3 million different strains of E. coli based just on those three antigens. Now, this is going to bother Rich Condit a lot. He had issues just keeping the nomenclature associated with spike. So how is he going to deal with E. coli? So Vincent, you got to ask Rich. We got to, and this is assuming that these aren't genetically linked. And then when you throw in the virulome into this, you can now appreciate how lucky we got with spike and the vaccine we were able to make against SARS-CoV-2. Next, they offered that they screen for virulence factors, or the virulome. Again, they found the virulence genes that I've already gone through. And they also offered, if that wasn't bad enough, that this strain, E. coli 141, from this poor 91-year-old woman, harbored several additional genetic clusters related to virulence factors. So you can well imagine that when this bacterium occupies a niche for which either the host has a limited microbiome potential to resist, our biggest defense mechanism is structure, namely our intestinal epithelial layer. And as we get older, it's not as flaccid and fluid as it once was. And so that begins to break down. So it also causes a problem for the treating physician in that they looked at the antibiotic associated genes that was associated with this. And at the end of the day, they went out on a global hunt. They characterized the genome of 104 sequence type 59 E. coli strains. All but four were serotype 01, flagella type number 7, and capsule type number 1. And it specifically had this KPSMTK1, which was identified in 86 strains of the 104. So when they began to look at the metadata available, it revealed that 89 of the strains had been recovered from people, 49% of which were isolated from the type of infection that killed this 91-year-old. 20 strains, or 22% of the sample that they looked at, were isolated from our gut, the gut microbiota, from a gut biopsy, and unfortunately no information was available. Again, this is in silico analysis. The authors also offered that not all E. coli need to express all of these pathogenic characteristics, but they only have to express enough to get that leg up. So it really drives home, or makes me begin to wonder if we are beginning to witness the beginnings where the paradigm will shift away from sole reliance on biochemical evaluation of microbes, or to a more in silico comparison to continuously assess not necessarily the risk to the patient, but the risk to the community, and then how best to address a treatment plan.
So in the clinical lab, should we get rid of chromogenic agar plates and just do sequence analysis?
I don't think we're there yet. I think we just don't have the throughput. In the clinical lab, the throughput, the ability to process the massive amount of samples that we have to get through. We need to rely on chromogenic medium. But the brilliance of this paper is that we had a curious tech who had MALDI-TOF data that said, this is E. coli, folks. And they looked at it on a plate and said, this is not E. coli. That's the only thing that can come from a human intervention. The ability to say, if it walks like a duck but doesn't look like a duck, can it still be a duck? And in this case, it was indeed still a mean, nasty duck. So summing up, this E. coli 141 strain, which was isolated from this bloodstream due to her gastrointestinal bleed, so meant it got out of her intestine, went into her body. She developed septic shock and she died. it was deficient in two critical enzymes that the clinical and public health microbiologists use routinely to detect, looking for E. coli. The only good news is the strains analyzed were susceptible to most of the antimicrobials tested. But the bad news was there was a PMRB mutation that results in colistin resistance. Colistin, remember, is the antibiotic that's used to treat gut infections as a last resort. And it was identified in 4 of the 104 strains that they tested. In concluding this wonderful story of this is a story of how microbes are literally on this continuous selective quest to kit themselves out for the great war against our immune system and the poor treating physician. And when I selected this paper, I really wanted to drive home the value that the clinical microbiologist has in providing to folks who think about pathogenesis that leg up to think, how are the microbes doing it?
The other thing, Michael, that really impressed me about this paper is not only did they study this EC141 clinical isolate, but they expanded it to so many other dozens of other strains from all over the world that were of the same sequence type, so the same kind of genetic fingerprint. And again, the vast majority of them, of course, didn't make the pigments on the standard indicator media, but they were also loaded with virulence factors. They could scavenge iron, they had capsule, they could adhere, they had a type 3 secretion system, they could make toxins. So we need to be aware that sequence type 59 is going to be lac minus, but it is virulent.
It's bad news. Bad news.
Could the reverse also be true? Could you find a salmonella or a shigella which can metabolize lactose?
Ooh.
Which goes against
dogma.
Against dogma.
Yeah, anything's possible with pathogenicity islands.
Mm-hmm. Michael, how did we miss this for so long?
I think we basically don't think about this. The price of sequencing has dropped like a stone. So I think more and more folks are sequencing. There's probably a number of clinical labs that do indeed miss it. And we don't have a quick and easy way to determine sequence type 59. Yeah. I'm hopeful that the diagnostic community will make a PCR primer that you can put into one of these blister plaques like BioFire or one of the others that could actually quickly detect whether or not, especially from blood cultures or urinary cultures, where it's an especially difficult situation for the treating physician.
So this work was led by a talented postdoc, Ana Carolina Damalo Santos. She is currently a postdoc in this lab with funding from the National Postdoc Program in Brazil. But she started out with a master's and a PhD in microbiology and immunology that she got at the Federal University of Sao Paulo in Brazil. And she is really appreciative that her PhD advisor, Rosa Silva, always encouraged her to pay close attention to everything that's going on, and also gave her the freedom to explore interesting observations that popped up. And clearly that the value of that approach came through in this study. So she also is very appreciative to be training now with Dr. Tanya Gomez, and says she now has the freedom to develop additional projects, including this one, which was in no way her main research project, but it's just something peculiar that somebody noticed, and they thought, let's follow it up. They started by, they were just characterizing E. coli strains from bloodstream infections. So clearly they knew that the strain was potent. And because once they saw this unusual plate phenotype, they followed it up, collaborated with people who could do the whole genome analysis, and appreciated early on this has practical implications, again, because we rely on these colorogenic media to screen our water, bodies of water, to make sure they're safe for swimming. So that is her story, and she is married. She has a 5-year-old son named Isaac, and she also shares that she was able to take almost 2 years off, starting when she was pregnant until her child got a little older, and then came back to the lab in her career in research when she was 9 months old, when the child was 9 months old. And Anna Carolina, I'll say I completely applaud that approach. I myself had both my kids when I was a PhD student, but I set up my postdoc so it didn't start for a year after I defended. So I had a year home with the 2 kids, and then came back and was delighted to be back in the lab and also have 2 wonderful kids at home. Mm-hmm. So her advice, and certainly a lesson that we all learned from this paper, is to be open to new discoveries.
Nice. Thank you, Michele.
Mm-hmm.
And thank you, Michael.
You're welcome.
Is this shaking the clinical microbiology community, Michael?
It should.
It should.
It should. I mean, they rely heavily on these automated systems. And in fact, at my institution, they no longer streak plates. They have an automatic streaker.
Yeah.
And, it's absolutely fascinating.
But they have,
go over and watch them.
They have mass spec, and you can easily do 20-minute identification. So it should be adaptable to this, right?
But you have to be able to grow it. E. coli we're still lucky E. coli we can grow.
Yeah.
It's the organisms that we can't grow.
Sure.
That become problematic because they're not in the MALDI-TOF database.
But this one can be grown. So
this one can be grown.
That's not a problem. Okay. All right. One email before we say goodbye. This is from Roy. This has to do with sourdough. Greetings. I recently heard about your podcast and was floored to hear you mentioning acetic acid bacteria in sourdough culture, when the fact is it only contains 2 critters, lactobacteria, homofermentative and heterofermentative bacteria, which produce lactic acid only and both acetic and lactic acid, respectively, and the wild yeasts. How did you come into the conclusion that vinegar bacteria participates in the sourdough fermentation? Vinegar bacteria, Acetobacter, are aerobic, and lactobacteria are often anaerobic. Can someone explain to me the biochemistry, how the vinegar bacteria participates in sourdough fermentation? As the vast sourdough literature does not mention it, I look forward to your reply. And so, Roy, we didn't come to this conclusion. The conclusion was the authors of the paper. And this is a paragraph from the paper that's relevant. They say, our integrative functional analysis of starter microbiomes highlights the disproportionate effects of acetic acid bacteria in shaping the sensory and leavening properties of sourdough. These bacteria have been historically underappreciated in most studies of sourdough microbes. So basically, Roy, we didn't notice that they were there. Now, Michael, his question about aerobic and anaerobic, how do you
it's all about substrate. It's all about substrate. Remember, the mission of a bacterium is to regenerate oxidized NAD. They'll dump those waste electrons to wherever they can go. And oxygen, when it's available, is the best place to dump waste electrons. You make water. However, if there's no oxygen available, you'll dump it to any organic acid, whether it be acetic acid, whether it be formic acid, whether it be lactic acid. You'll dump them because you can't eat a sugar unless you have oxidized NAD. And that's probably what's going on in this beginning fermentive process, where you're accelerating that drop in pH, and the acetogens, the ones that will make acid, drop it to a low enough pH where the lactic acid bacteria then can predominate, and you don't get those damn yeasts coming in ruining the flavor of the bread.
Got it.
And Vince Young probably has it all mapped out down to sequence using some of Pat Schloss's fancy tools. And he probably knows every microbe in his starter culture. And that was the picture that Michele showed us.
Yeah,
so
actually,
Michele, you got to talk to Vince.
Yeah, I don't, to my knowledge, Vince is not looking at the microbiome of sourdough. He's got bigger fish to fry right now, focused on C. difficile and other infections. But
he has children who need science projects. I met one of his sons at an ASM meeting. He can assign this project to his son.
yeah, I have to tell you, they're grown.
That's the problem with kids. They grow.
Michele, did you make some bread from his starter?
I did, and I have some with me this week. It's delicious.
Is it really good? Huh? Cool.
Yeah, it sure is.
All right. That is artisanal food, right, Michele?
You bet. Craft
food.
Beverages.
All right. That's TWiM 244. You can find the show notes at microbe.tv/TWiM. If you'd like to send us a question or a comment, you can do that to twim@microbe.tv. And if you like what we do, consider supporting us. Microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
My pleasure. Thank you.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure.
And Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Michael, you ever make sourdough bread?
I tried. I failed. The fermentation, the yeast ended up overwhelming. I think it's too warm here.
Yeah. Try it in the winter.
Okay.
Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. I really like this food arc that we have. We have to keep it going because we had beer today, or a fermented beverage. We had bread.
We had steak.
We had steak.
Beef.
Now, did you get the delivery of Omaha steaks? I'm just curious and not tell us.
No, I don't get them. We used to years ago, but I stopped eating beef, man. I can't. I got to stay away from red meat. It's not good for me. With my family history. I would like to thank the American Society for Microbiology for the support of TWiM, and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.